undergraduate honors thesis deals with exploring the role that ZNF217, a zinc finger protein, has in the role of carcinogenesis, especially in osteosarcomas, also known as cancers of the bone, and its potential capability of being a target of a chemotherapeutic drug called tricirabine in osteosarcomas, as well as other cancer types. A little background on ZNF217 is a transcription factor that has previously been shown to be a candidate oncogene that interacts directly with ERB3, a member of the epidermal growth factor receptor family of receptor tyrosine kinases. Downstream pathways of ERB3 have important implications in the control of the cell cycle and proliferation through activation, or in this case the hyperactivation, of the PI3K AKT pathway, leading to unregulated cell growth. If this growth is left unchecked, cancerous cells with the ability to metastasize to other areas of the body may arise. My thesis has two main goals, the first being to further functionally characterize ZNF217 in several osteosarcoma lines with and without the treatment of tricirabine, as there is no previously published data specific to osteosarcomas. There is, however, functional data on a handful of breast cancer cell lines that was collected by Lori Littlepage's lab at Notre Dame, where they showed an increase in colony counts in an anchorage independence assay, as well as increased migratory ability in a scratch healing assay when ZNF was overexpressed in her cell lines. They then showed a reduction in these values to below the basal level when these cells were treated with tricirabine. I plan to model my experiments closely to those found in the Littlepage publication so that I will be able to have some kind of reference with which to compare my results. The second goal of my thesis work is to attempt to characterize the mechanism by which ZNF217 utilizes to manifest a cancerous phenotype, as well as where tricirabine fits into this mechanism in order to lessen or completely eradicate these cancer cells. Dr. Littlepage did propose a mechanism in her publication, but even hers contained unknowns and question marks, which I hope to uncover the answers to. There are many molecules and states of these molecules, either active or inactive, within these interconnected pathways that are able to be detected and quantified. I'm optimistic in my chances to find some missing piece that may be of some value in understanding the actions of ZNF217 through treatment of tricirabine along with the combinations of other inhibiting drugs. The potential significance of this project could be great, as osteosarcoma cell lines are extremely heterogeneous, even by cancer standards, which makes them very difficult to treat with specific chemotherapeutics. In an attempt to bypass this heterogeneity, current prescribed treatments include combinations of more general chemo drugs, with the hope that the patient's tumor will respond well to at least one of the drugs in the cocktail. If elevated levels of ZNF217 were able to be shown in a biopsy of the patient's tumor, then tricirabine could be selectively added to those patients' chemo regimen in addition to the other general drugs, hopefully increasing the chance of relapse. As osteosarcoma is a cancer of the bone, it is understandably found most in children and elderly adults, as their bones are quickly growing or slowly deteriorating respectively. Any significant improvement in the patient outcomes due to detection of elevated ZNF217 levels and the subsequent addition of tricirabine to their treatment could hopefully extend the lives of these patients, or at the very least improve their quality of life.